Hello everyone, my name is Kendrick Yim. I'm currently a fourth-year pharmacology major here at UCSB. And this summer, through the Beckman Scholars Program, I've been working in the lab with Dr. Joel Rothman in the Molecular, Cellular, and Developmental Biology program under the mentorship of Dr. Nate Dudley and Dr. Pradeep Joshi. The title of my talk today is Exploring the Effects and Mechanism of the CEP1 tumor suppressor on chromosome non-disjunction in C. elegans. So cancer, in its most basic definition, is unregulated cell proliferation, eventually forming large masses of cells called tumors. These tumors can then grow and metastasize and ultimately inhibit normal cellular function. The American Cancer Society estimates that 1 in 2 men and 1 in 3 women will actually develop cancer at some point during their lifetime in today's population. However, even more worrisome than this statistic is that cancer mortality rates have remained stagnant in the past half-century. So while tremendous progress is being made in other diseases, such as heart disease and cerebrovascular disease, if you look at the death rates in cancer from 1950 to 2002, they're almost identical. So in order to improve these numbers right here, I think we need to take a step back and analyze more closely the foundations of cancer formation and pathogenesis. So to begin with some background, P53, also known as the master guardian of the genome, is a tumor suppressor. And tumor suppressors are gene products that normally protect cells from mutations that would otherwise allow it to become cancerous. In fact, P53 is the most commonly mutated gene in human cancers, and it is nearly impossible for a normal cell to transform into a cancerous cell without actually inactivating that P53 network in some way. So a fully functional P53, once it senses genomic damage, can activate one of 2 pathways. The first being a cell cycle arrest pathway, thereby giving the cell more time to actually go in and repair that damaged DNA. Or if the damage is too great, it can activate programmed cell death, thereby just eliminating the damaged cell and DNA. And the whole point of this is to prevent the accumulation of mutations and basically maintain genomic stability. However, a form of genomic instability and one of the hallmark features of cancer is aneuploidy, or an abnormal number of chromosomes. And there's a direct correlation between cells that display aneuploidy and cells that have an inactive or non-functional P53 tumor suppressor. And one of the ways that aneuploidy is achieved is through an event called chromosome non-disjunction, or basically improper segregation of chromosomes. So I've simplified this concept in the diagram here. So suppose this is a gamete progenitor cell, or the cell from which all your sperm and egg cells are derived from. I've simplified it down to 2 copies of 1 chromosome. And during meiosis, you expect equal segregation of the chromosomes. And if you continue down this normal pathway on the right side, you end up with 1 chromatid in each of the gamete cells. However, if you have a non-disjunction event, 2 of the chromatids can actually segregate together, leaving 1 gamete cell with 2 copies and the other with none. So the question that I'm looking at is, does P53 play a role in regulating these non-disjunction events? So specifically, I'm going to be studying the CEP1 tumor suppressor in C. elegans as a P53 model. CEP1 and P53 are homologs, meaning that these 2 tumor suppressors actually evolved from the same parent protein. And we use C. elegans because it provides us a plethora of genetic and biochemical tools. But most importantly, it's easy to assay for X chromosome non-disjunction. So C. elegans has 2 sexes, hermaphrodites that have 2 X chromosomes and males that have 1 X chromosome, the other one being lost from a non-disjunction event. So by simply comparing the proportion of males versus hermaphrodites, one can actually quantitate how much X chromosome non-disjunction is occurring. And in addition, the DNA binding domain between CEP1 and P53 are highly conserved. In fact, the arginine residues that actually contact the backbone of the DNA are completely conserved, and the 2 tumor suppressors can actually recognize the same DNA sequences. So the ultimate goal of my project is to explore the effects of knocking out and overexpressing CEP1 on chromosome non-disjunction and to figure out how exactly this is occurring. So in the first part of my experiment, I looked at the effect of CEP1 on the X chromosomes, or your sex-determining chromosomes. In order to do this, I generated a heat shock CEP1 Hem5 string. And what this means is I took a hermaphrodite that has a CEP1 gene fused to a heat shock promoter. So when these worms experience elevated temperatures, they'll actually overproduce the CEP1 protein. And I crossed that with a Hem5 minus male, and what this means is that these males are lacking the Hem5 gene. So they can't produce the Hem5 protein, which is normally necessary for regulating X chromosome crossover and distribution events, which is why we see such high levels of X chromosome non-disjunction in the first place. So once I've confirmed I had the right strain, I heat shock them at 33 degrees for half an hour during gametogenesis, or when their egg and sperm cells are being generated, and compared the percentage of males and hermaphrodites in the progeny both before and after the heat shock. So to give you an idea of what I'm looking at exactly, in figure A we have an adult male, and in figure B we have an adult hermaphrodite. And upon first glance they look pretty similar, but the distinguishing feature that I'm looking at to differentiate the two is the presence of a male tail here, which is a thicker arrow-shaped tail that the male uses for mating that the hermaphrodite lacks. In the second part of my experiment, I looked at the effect of CEP1 on autosomal non-disjunction, or your non-sex chromosomes. And in order to do this, I generated two different types of strains. The first being a HIM6 heat shock CEP1 overexpression strain, and a HIM6 heat shock CEP1 knockout strain. And we utilize this HIM6 background because these worms, again, are lacking the HIM6 protein. And this HIM6 protein is a DNA helicase normally necessary for DNA recombination, DNA repair, DNA replication. So in this background, we actually see high levels of autosomal non-disjunction, and consequently, we see high levels of embryonic lethality, since these organisms cannot survive without a full complement of their autosomes. However, there are some special cases in which you can have autosomal non-disjunction without embryonic lethality. And without going into the details of this, we can actually place phenotypic markers such as dumpy or uncoordinated on certain chromosomes, and based on the presence or lack thereof of these markers, you can actually identify certain autosomal non-disjunction events. So from my experiments looking at the effect of CEP1 on the X chromosome, what we found was that overexpressing CEP1 actually suppresses the HIM phenotype. So I've included a lot of my control data here also, but I've highlighted the two most important parts. So on the y-axis, we have percentage of males, and in the HIM5 strain alone, in this first blue bar, we saw about 36% males. After we created the heat shock CEP1 HIM5 double mutant, we saw that decrease to 26% after heat shocking them for half an hour. So ultimately, overexpressing CEP1, we saw a 29% decrease in the proportion of males, or alternatively, a 29% decrease in the X chromosome non-disjunction. So what do all these bars mean? Ultimately, we've identified a new role of CEP1. Basically, CEP1 is regulating X chromosome segregation. More specifically, it's actually suppressing X chromosome non-disjunction. So in the absence of HIM5, CEP1 is being recruited, or recruiting other elements to help facilitate proper X chromosome segregation. So currently right now, I'm trying to determine the optimal window for CEP1 suppression within that 18 hours of gametogenesis. I'm also going to do a qPCR to quantitate levels of CEP1 expression, and I'm finishing constructing my HIM6 mutants, and we will begin to score them shortly. And hopefully by the end of this project, I'll have some time to figure out the exact molecular mechanism by which CEP1 is functioning. So by increasing our understanding of CEP1, may translate directly to increasing our understanding of p53. In many aspects, CEP1 is analogous to p53. They have a similar structure, they have a similar function, and they can even recognize the same DNA sequences. So if CEP1 has this role of regulating chromosome segregation in C. elegans, then p53 may also have the same role in humans. So by completing these experiments, I hope to provide a more thorough understanding of p53's role in cancer pathogenesis, and even perhaps provide a new target for more effective diagnostic and therapeutic tools in the fight against cancer. So lastly, I'd like to thank Dr. Rothman for allowing me to work in his lab, my two mentors who are sitting in the back, Dr. Dudley, Dr. Doshi, in addition to all the other Rothman lab members, Erica, Maria, and Tyler for putting this program together, and last but not least, the Arnold Naval Beckman Foundation for providing the funding through the Beckman Scholarship Program.